So everybody in class has already done the eyeball dissection. So this is old hat stuff. You guys are all familiar with this. So let's review the cartoon picture here together quickly, and you should be very familiar with all of these structures. Then we'll go into the notes. We'll fill in some spaces in the notes and just keep plowing on through this material. Structure of the eye. Well, it's a globe, and here the notes say eyeball is a hollow sphere filled with fluids called humors, and that's what we do. We use the Latin term for, there's always somebody at the very last. We use the Latin term of humors for fluids. So we had two main types of fluids inside the eyeball. One was called the aqueous humor, the other was called the vitreous humor, and they are very specifically located in certain areas of the eyeball. We have a name for two distinct areas that are kind of a large type of areas. We can either call them segments, or in your text it calls them cavities. So we have a posterior segment or cavity and an anterior segment or cavity. And they used to be called segments in our previous text. Where is the vitreous humor? Posterior segment or cavity. We got another latecomer coming on in. I should have given just a few more minutes before I shut the door. So the anterior cavity has the aqueous humor, and what's unique about the anterior cavity? It's subdivided into chambers. We have an anterior chamber and a posterior chamber. What's the dividing line between the anterior chamber and the posterior chamber? The iris. The dividing line between the anterior cavity and the posterior cavity? The lens. Okay, so that's, you know, really simple things to do whenever you guys are doing any sort of eval. It's always good to go to your lecture notes, by the way, and take a look at what your lecture notes want you to know, because I bet the lab will reflect what we want you to know in lecture. So don't forget that. These things are listed here, and if you would have gone to this diagram and started to make a list of things, it would have helped you quite a bit in your eval. Now, we have layers to the eye, and we call those layers tunics. And there are three tunics to the eye. There is a fibrous tunic, a vascular tunic, and then the retina, which is a neural layer, a neural tunic. The fibrous tunic, the bulk of the fibrous tunic is made up of sclera, dense irregular CT, right? It is really tough, and you guys saw how tough it was when you had to actually incise it and try and cut around it, correct? That's some of the toughest material you've had to cut through in a long time in A&P. So that is really, really tough material. As a matter of fact, that material is, the sclera itself is an extension of our dura mater. You guys remember the dura mater? And remember how tough that was? So that's what's really fascinating. Our sclera is basically an outward extension of the dura mater. Now, the fibrous tunic is not done with just the sclera, because there's another small area of the fibrous tunic that is very different in its appearance. And that's because we have actually changed the arrangement of the fibers in that area, and that area is called the cornea. The cornea is a clear, transparent region where we've actually made the fibers very regular in that area, and that's also part of what helps us to transmit light. They're also indeed very, very densely packed to allow for that clearness, that clarity that we actually see. And we'll talk a little bit more about that too in a moment. So those are the two main regions of the fibrous tunic. Now we go inward and we find the vascular tunic. The vascular tunic, the bulk of the vascular tunic is called the choroid. But the vascular tunic is more than just the choroid, right? When we move anteriorly, the vascular tunic becomes the ciliary body, as well as in front of that, our iris. So all of those are part of what we call the vascular tunic. So remember, when you take a look at the notes, we actually have them organized in a certain column. So our fibrous tunic, if you take a look under here, fibrous tunic is one. We have the sclera and the cornea subdivided in that area. So same thing happens in our note construction. That's the outline that we actually have. Now, with the ciliary body has the ability to actually help us change the thickness of our lens, and we'll get into more details on that in just a minute. But it has projections, and what do we call projections in the land of bones? Processes. So ciliary processes, they're obviously very small, but off of the ciliary processes, which are smooth muscle, are actual little lines, little fibers, and they're suspensory ligaments or ciliary zonules, right? So you see them like that in your notes. So you mentioned all of these things that I'm talking about right now was something that you were going to mention in your eye eval. So that's attached to the lens. It's actually attached to a capsule around the lens. And one of the things that you have to remember is when you have an organ has a capsule. Even something like the lens, there is a capsule, and that's a very important thing to note. Otherwise, we wouldn't be able to do certain surgical procedures without that capsule being there. And that surgical procedure is actually or cataract surgery or replacement of the lens. Now, how many of you were able to actually see the fresher specimen, not the preserved specimen, the fresh lens? Okay. That was really great. The first Monday labs, we forgot to pull them out of the freezer, so you would have never been able to appreciate their consistency at all. But a fresh lens is not at all like a preserved lens. A preserved lens is like, to me, it's like as hard as a marble, so to speak. You know, it has been denatured, just like you take a raw egg and you fry it. I mean, it's just not the same as the raw product. And so it's really hard for those of you that did not actually see this in lab, and maybe you can ask your lab instructor to pull out one of the fresh specimens out of the freezer and let it thaw out for one of your labs to see what a real lens looks like, because it is gelatinous. Just like the vitreous humor was gelatinous, the lens is gelatinous, not quite to the degree of the vitreous humor, but it is very gelatinous. It's squishy, mushy. It is pliable. And that is something that you do not appreciate with our preserved specimens. The formalin takes that completely away. And that's why I really appreciate one of our students. Her husband has been collecting these eyeballs for a while, and they've been freezing them and giving them to us. And so I said, yes, I'll take them. So the bigger the eyeballs I can get, the happier I am. So if anybody knows anybody that's going to be hunting elk or moose or something like that, and you know, say, pluck those eyeballs out of that animal and save them for me or save them for you. Either way, it's actually very fascinating to take a look at a real fresh specimen. So the lens is very pliable, held in place by the suspensory ligaments or the ciliary zonules, and not just those guys, but the vitreous humor. You guys, some of you had the lens stuck to your vitreous humor. Yes, some of you did. And so you have to realize part of the reason that the lens stays in its place is because of the vitreous humor. You will also hear me call the vitreous humor the vitreous body because it has form. It actually has a shape, and is actually what is responsible for giving our eye its circular shape and to keep it, the retina applied to the choroid. Because the retina, you guys noted, was not attached except in one spot. Optic disc or the blind spot. So that's the only true attachment site of the retina. So the retina is not attached to the choroid. That's the inner layer, the neural layer. And it is, when you poured out the choroid body, some of you, it collapsed right away. And that's an example of retinal detachment. And so when somebody has retinal detachment, they're having a problem with the vitreous body. So lab and lecture do correlate. And so take what you learned in lab and learned in lecture and keep those images fresh in your mind with you as you study. Now, the ora serrata listed here, that's something that we like to have students name when they do their eval. That is the edge of the ciliary body. So ora serrata, ora is oral or round, and serrata, I hope you can relate to serrated. And so that ora serrata was at the edge here. And you know what else was at that edge? The ora serrata, the ciliary body, edge was there. And remember, it's part of the vascular tunic. But let me ask you, how far did the retina go? Did it go all the way around your it stopped at the ciliary body. So the retina actually stopped at the ciliary body where you see that ora serrata. And so that's another manifestation of that area. Now, we're going to talk about aqueous humor or aqueous humor, doesn't matter how you say it. It is produced by the ciliary processes of the ciliary body. And what's interesting about that, it is produced 24/7. What else is produced 24/7? A clear fluid. Cerebral spinal fluid, and it's just like cerebral spinal fluid, except it does have a lot of nutrients also inside of it, because these nutrients from the aqueous humor are actually going to help feed the cornea as well as the lens. So, because they have no blood supply, right? The lens, did it have any blood supply going to it? No, and it's still a living structure. In order for it to be nice and flexible and gummy bear-like, it has to be alive, and it has to have a nutrient supply. And so your aqueous humor is actually supplying nutrients to your own lens. It is also helping to supply nutrients to your cornea. So it's not just a fluid. It is a nutrient broth for areas of your eye that are avascular, and such as the cornea and the lens. Now, if it's produced 24/7 in the posterior chamber, where do you know it also can be found? In just the posterior chamber? Anterior chamber. And the reason it gets into the anterior chamber is because there's a hole. It's called that pupil. And so it gets into the anterior chamber via the hole, and it's produced 24/7. Well, if you produce it 24/7 and there is no outlet for it, what happens to the fluid pressure inside the eye? It goes way up. So there is an overflow valve. Just like CSF, we had the overflow valve in the arachnoid villi and arachnoid granulations. We have an overflow valve for our aqueous humor, and that's at the scleral venous junction. So the scleral venous junction right here and right here is where we have the sclerocorneal junction. Sorry, people. Sclerocorneal junction. You'll find the scleral venous sinus. Now, this is a sagittal section through the eye itself, and so you have to realize that this is a slice through something that is circumferential. So the scleral venous sinus is circumferential, so it goes all the way around the sclerocorneal junction, and it's an overflow area. Just like sometimes most swimming pools have an overflow gutter for the pool water to flow into, same thing here. And so if that gets blocked, then you experience a phenomenon called, in us, glaucoma. Okay, so glaucoma is what I would kind of call like high blood pressure of the eye. You guys kind of follow me? It's not really blood pressure, but it's high pressure of the eye. So synonymous to high blood pressure. Is high blood pressure good for you? Is glaucoma good for you? Do you guys know what happens to people who get glaucoma? They go blind. And the reason they go blind is it's an elephant sitting on a piece of wet toilet tissue. Right? Because the retina is essentially synonymous with wet toilet tissue. So you're going to smush it and you're going to kill all of your photoreceptors. Once you kill your photoreceptors, do they come back? No. And so that's why as we age, we have more issues with the drainage related stuff. You know, time does have an effect on all of us. And so when they go to the eye doctor, most people under ordinary circumstances, if they go to the eye doctor on a regular basis, they're always checking their eye pressure. So they check their eye pressure because if the eye pressure starts to get higher, then they know that the pressure on the retina is increasing, and then the chance of actually killing off your photoreceptors is increasing, and therefore chance of blindness is increasing from that process. So that's why the scleral venous sinus is such an important area to be operating properly. And so they have medications that help to keep it patent, open, or dilated. Because as we all age, things do get stiffer and actually get a little blocked up. And so medications can be used to help out with that. So I think that's most of the picture there. Let's go ahead now and look at the note portion. So tunics or coats form the wall of the eye, and there are three of them, 3 layers. The first outer layer, the fibrous tunic, is avascular, dense fibrous CT, and you know it's regular, because if it was, it's irregular, because if it was regular, when somebody sucked you in the eye, it would split like a banana. So it's dense irregular. So if I said regular at any time, you know, I was just having a brain fart moving too fast. Okay, of the fibrous tunic, the bulk of the fibrous tunic is the sclera, which is the white of the eye, and it is designed to protect and maintain the shape of the eye. It is continuous with the dura mater. It is the toughest part of the eye, and it covers the optic nerve. As we move anterior on our fibrous tunic, we come across that cornea, which is nice and clear. And our cornea is interesting because, unlike the sclera, contains many pain receptors. And you can kind of understand that. We don't want damage to occur. So you're going to make sure this area is actually rich in pain receptors so that the area that allows you to see is really protected from things that can traumatize or damage it. So that's a protective mechanism. You know, pain receptors are put in areas where you need more protection from those things. We have less pain receptors on our back. We have more pain receptors and other sensory receptors on our ventral side of our body. So wherever you need more protection, you'll have more pain receptors. Our structure of our cornea is convex, modified, transparent part of the fibrous tunic is the descriptor. So you have to memorize convex and concave. Now, this is what I do. You can find your own method, and if you want to post it on the forum, I'd be very happy to have you do that. I am vexed when my belly protrudes. Convex. And whenever you go into a cave, you go in. So concave is in, and concave is what vexes me. So convex. So that's the way I memorized it. So yeah, there's always tricks to memorization. And there's nothing wrong with actually using those tricks. What's fascinating is we do have a protective layer over that cornea, and it's a stratified squamous epithelium. And remember, stratified squamous epithelium is designed for protection and trauma and friction and things like that. So it's interesting. We have conjunctiva over the sclera, but we don't have it over the cornea because we don't want conjunctivitis where we need to see. And so what we do is we have a very transparent layer of stratified squamous epithelium covering that. And indeed, because it's stratified squamous epithelium, we know it's protective, and we do have some sloughing of the very surface aspect of it, but it's not important because it's stratified and we're always getting a continuous replacement of that. Realize that this joins the conjunctiva at the edge of the cornea. So we have protection actually all over the exposed surface of our eyeball itself. Now, when you get corneal abrasions, this is the area that is damaged. Make my eye go away. So this is the area that's damaged. And remember, I said there's lots of pain receptors. When people get corneal abrasions, it hurts a lot. And they want to keep their eyes closed. And you know that if you keep the area protected, the nice thing about stratified squamous epithelium is it gets replaced. So if you rip some off, you rip some of your skin off, and if you leave enough of a base of your skin, you know that you will get normal skin to actually grow back. And that's what a corneal abrasion is, and that is why they're so painful, because it's designed to keep your eyes shut so that there is a nice moisture bandage over them. You guys follow me with that? Nice protective moisture bandage over them, and so we can re-epithelialize. So that's an interesting phenomenon for our cornea. Interestingly, so now we have our lovely fibrous CT that's actually linear and regular underneath that stratified squamous epithelium. And then when we go to the inside of the eye, we also have squamous, but here it's just simple squamous epithelium on the inside of our cornea. And the interesting thing about this, these cells contain sodium pumps. And the sodium pumps are to pump sodium out of the lens. Oh, it's not lens, excuse me, cornea. We also have them on the lens. So we want to pump sodium out of the cornea. Now, sodium is synonymous with salt as far as I'm concerned. Yes? Okay. And what do we always know wants to go where salt is? Water. So this is very important. These simple squamous cells on the inside of our cornea are making sure salt does not infiltrate the cornea. Because if salt infiltrates the cornea, so will water, and then the thickness of the cornea will change. You guys follow me with that? And so that is going to affect your visual clarity, because one of the main structures responsible for refraction of light is actually your cornea. Your corneas are primarily responsible for refraction or bending of light. We'll get into that a little bit more later. But that's the main structure that's going to bend light. Our lens is going to help us with what's called near vision. And so it's more important for that aspect, or bending light, to help us with things up close to take a look at that. But the cornea is a major structure for the bending of light to make sure it hits the sweet spot so we see well. Now, when people get diabetes, what level is up in their blood? Sugar, glucose. Well, is it only their blood that experiences this glucose overabundance? So the other thing that happens when people get diabetes, their blood glucose level goes up, and it's typically up for a long time for adult onset or type 2 diabetes. And they start to complain of visual problems. You guys following me with this? So what I'm telling you too is not only does the salt want to go into our cornea. If we have too much sugar in our blood, we'll end up with too much sugar in our aqueous humor, which means we'll end up with sugar trying to go into the cornea, which means this is another osmotic thing that's going to pull water into it, and they will get a thickening of their cornea and a distortion of their vision. So that's why we have some of these phenomenons happening, because of the elevated sugar is also reflected in their aqueous humor. So
diabetes, I know like a simple eye exam, you can see, diabetes. What are they looking for with that? What do they see?
Well, they see all kinds of deposits actually starting to occur on the surface of their retina for diabetes. But you can look that up and take a look online and see what a normal retina looks like, and then what a diabetic retina looks like, and you'll see the changes with that. So that's out there, but we don't want to go into that too much right now. So pump sodium out of cornea for clarity. Because as you saw with the cow eye, how'd the cornea look to you? It was no longer clear. So the pumps weren't working. Sodium was able to actually infiltrate as well as water, and we had actual distortion of the clarity of the cornea. I'm on the wrong word track today. So if I keep doing this, you guys stop me and say slow down on that. The other thing too about the cornea, it is avascular. So it means it does not get a blood supply. So it's real important for you to also realize the aqueous humor inside is providing nutrients to our cornea. So the aqueous humor is a nutrient broth to help feed that. The other thing too is that oxygen and carbon dioxide are actually able to diffuse across the cornea freely. And in the days of old, when they first started making contacts, what kind of material do you suppose they used to make contacts out of? Glass. How about glass and diffusion? And in the days of old, when the first contact users wore contacts, they were made out of solid material like glass. They put them on their eye, they covered up their cornea completely. And what happened to the diffusion of oxygen and carbon dioxide at that stage? So they were only able to wear their contacts for a limited number of hours in a day. And so then they came up with still rigid lenses that were called gas permeable. You now see why they created gas permeable lenses, so that you could then wear gas permeable lenses all day long and not have to worry about taking them off to actually let blood supply back to the oxygen supply back to the cornea. It's like putting a rubber band around your finger all day long. You know, after a while you're going to have a bad result. And so they had to take those hard lenses off because they were actually stopping the diffusion of oxygen to and through the cornea, and it was causing great harm. So avascular, no blood supply, and also not accessible to the immune system. And this is absolutely fabulous for another reason, and it's called transplants. You can use these guys and you can transplant the cornea of one person onto the cornea of another person and not have rejection-related issues. Avascular and not hooked up to the immune system. So a lot of people, when they pass away, they actually donate their corneas. And so they will come in and they will literally shave off an individual's cornea, and they will put these little cups over the eyes so that the eyes don't fall back in, so that when people have funeral services and stuff, they often have viewings of the loved one that has passed away, and so they don't have some weird looking depressions in there which freak people out, you know. They think they're off to some horror movie or something like that when they look at their deceased one, which is already looking kind of scary to them in the first place. So that's what's really cool. They take those corneas, they freeze them, and then they actually shape them. They can grind them down and stuff along that line to actually create that kind of surfaces that they then will apply to another person's cornea, which they have roughened up so that they sticks on, and then they actually have a difference in thickness of their cornea, which is the major area where refraction or bending of light actually occurs.
My nephew died at the age of 26 of brain, and he fell. So my sister, harvested his body. My nephew was going blind though. So he had no peripheral vision, but they were able to transplant his corneas because there was nothing wrong.
It was nothing to do with the corneas that was affecting his vision, correct?
It was
Yeah.
pretty crazy that they could use his eyes for anything.
I know. So there's understanding the construction of the various aspects of the eye is very important in understanding how they actually work so well for us. Now let's go inside. So that was our fibrous tunic. We're now going inside. The next layer in called the vascular tunic. Also another term that you will see out there is uvea. UVEA for the vascular tunic. That is the middle coat of the eye of the three tunics that we were talking about, and it has three regions. The choroid, which is the bulk, then as we move anteriorly, the ciliary body, and then in front of that, the iris. So let's take a look at that choroid, and it's in the posterior cavity. Feeds the fibrous tunic. Because it has very poor vascularity associated to the sclera, and it is full of dark brown pigment. And this is important for us because we want to prevent light scatter. When light scatters with our retina, it activates other photoreceptors, and it will greatly impact the clarity of our vision. Now, the tapetum lucidum in your cows and dogs and cats and things along that line, that is designed to actually promote more light scatter. Because what that does, that allows them to see better in low light situations or in more darker situations. So we all know other animals see better in the dark than we do. And that's because our eyes are designed for daylight function. And their eyes are actually designed to help them at night, because a lot of them are hunting at night and things along that line, or being hunted at night. And so they want to be able to see what's going on around them at night. And that light scatter allows them to see objects in the dark, but their clarity or the crispness of the edges of whatever they're looking at is definitely less than ours. So they see in the dark, but they don't see as clearly in the dark. We see so very clearly in the light, much better than they actually do because of that phenomenon. The ciliary body going anteriorly is continuous with the choroid. It is mostly smooth muscle. Its processes secrete the aqueous humor. Suspensory ligaments come off of the ciliary body's process and attach to the lens capsule edge. And help hold the lens in place. Plus, there is more to that story because they also affect the thickness of the lens. And we'll talk about that in just a little bit. The iris, that's the visible colored part of the eye. Anteriorly, it is shaped like a donut. The hole inside that donut we know is the pupil. And the color of the iris is directly related to the amount of dark brown pigment.
Is that related to the dark brown pigment in the choroid too?
No. When we're born, the choroid have an abundance of dark brown pigment, but when we take a look at the iris, the amount of dark brown pigment changes. It is the least when we are born and then increases as we age, and around 2 or 3 years of age, then it's kind of, what it's going to be for the most of your life. And then as you age, you start to lose some of that. So people's irises actually get a little lighter for many of them as they age. Hang on, let me finish this. Color directly related to amount of dark brown pigment within it. Infants, Caucasian infants, do not have much when born. As age, this accumulates. And then we see the actual color of the eye that we will say that we have when we say we have blue eyes or brown eyes or green eyes or hazel eyes later on in life. So for Caucasian infants, they're born with very little dark brown pigment, and the color of their eyes are almost always blue. Almost always blue. There's always an exception to the rule, but I'm on the general path here, and there's always those exceptions. That's why I also specify Caucasian. And so what happens is there's not much dark brown pigment, and there's the muscle fibers as well as vascularity associated with their iris. And as they age, more dark brown pigment is being produced based upon their genetic profile, and they'll continue to produce more and more dark brown pigment based upon what they are genetically destined to have. And ultimately around age 2 or 3, you'll be able to say whether your baby has blue eyes or green eyes or brown eyes. I had a friend whose daughter insisted that her brown-eyed baby had blue eyes because she wanted her son to have blue eyes. And when he was born, he did have blue eyes. And she never saw anything but blue after that. We're all just going, kid's got brown eyes, sorry. Because that baby was born with blue eyes. And indeed, that is the case. Now, when you have a complete absence of pigment, what color is the iris? Red. So there is some, and that's why you have the blue. But if there is an absolute absence and albinism, then it's actually red. To add to our notes, just pupil in center is the opening. Just add it to the notes. And size of the pupil is controlled by the iris. The iris is muscular, and it is smooth muscles because is it under voluntary or involuntary control? Involuntary control. So remember, things that are involuntary are, other than the heart, are basically going to all be smooth muscles. So it is smooth muscles, and the smooth muscles are arranged in a radial pattern as well as a circular pattern. So we actually have a radial direction to those smooth muscle fibers as well as a circular direction to those smooth muscle fibers. And when you contract the circular smooth muscle fibers, what will happen, without looking at this picture and just thinking about what will happen to the size of the pupil, gets smaller. So the circular muscle fibers will actually make it smaller, and we all know the pupil does get smaller in bright light. And then when it's dark and we want to get more light in to see what's going on with what's going on around us, we want that pupil to open up or dilate. So you relax the circular muscles, correct? And therefore contract the radial muscles, because the radial muscles will pull things off to the perimeter. Now, and that's indeed what we find in low light situations, that dilation. It's important to figure out a way to link sympathetic and parasympathetic to which muscles are being contracted. And sympathetic, remember, is fight, flight, right? Parasympathetic is rest, digest. So I want to ask you, will you see more of the world with a dilated pupil or a constricted pupil? Dilated pupil. And if you're running from the bear, do you want a dilated pupil or a constricted pupil? Dilated. Okay, so that's how, again, these are my methodologies in trying to help me to remember this without having to memorize everything. Because memorizing everything is too hard, people. I don't know why you guys want to do it that way. I prefer conceptual learning to memorization. Okay, I memorize a few things and then I apply concepts to everything else. That works so much better for me. So in our notes here, it says the radial are controlled by the sympathetic nervous system. And remember, that makes the pupil larger. That makes sense. And the circular ones are controlled by parasympathetic nervous system, which makes it smaller. When it's smaller, we're also going to talk about that's a part of the accommodation process. Things get closer to you and you're not afraid of them. So it's okay to get nice and small as things that you're not afraid of actually get closer to you. So The sympathetic, just realize if you can remember one with a process, then the other is the opposite. Now the retina, our innermost tunic, has also two components to it. Its outermost component, which is adjacent to the choroid, is the pigmented layer. It's very thin. This pigmented layer of the retina is very thin. And it is also there to help absorb extraneous light. We want to prevent light scatter because we want to only stimulate the photoreceptors that the light is aimed for, not to have overflow. And it is also important because this is where vitamin A comes to our photoreceptors. You may have heard vitamin A is good for your vision, and it is. Vitamin A is necessary for healthy photoreceptors, and it is produced in this pigmented layer, which the photoreceptors are actually buried, their little tips are buried into this. So they can get their vitamin A directly so that they can actually undergo the restorative process that they need to do in order to actually function properly. If you know anything about worldwide poverty, one of the problems with worldwide poverty or areas in the world that are poor is proper nutrients. And without proper nutrients from a proper diet, there are large areas of the world that have a problem with blindness. That is nutrient or dietary only. And that is because they are not getting dietary precursors to actually form the vitamin A for their eyes. And so this vitamin A is an essential nutrient for healthy rods and cones. And there's actually areas of blindness for this. And so they're also doing GMOs, genetically modifying crops, to actually create precursors to vitamin A in the diet. You know, like carrots and carotene and things like that. Those are all precursors to vitamin A that once we ingest them, we can actually utilize them to actually make vitamin A. And so there's been an initiative, and I don't know how far they've gotten, in creating what is called yellow rice. And the yellow rice is genetically modified rice that they're trying to get out to those areas of the world where rice is a staple. And there's a lot of areas of the world where rice is a staple, yes, but they don't have much else. Rice and beans and things along that line. So they're trying to create these crops that people can grow and to provide the adequate amounts of vitamin A in their diet so that they actually have good vision. So neural layer, we're going on in. That was a pigmented layer, very thin. Now the neural layer is interesting because when you guys took a look at your retinas and your specimens, were they transparent? No. Again, the preservation processes changes what the real living organism and structures are. So it's a transparent nervous layer, and as I want to relate it to, it's an out pocket of the brain. So I liken, my hands are coming out to you as these are optic nerves coming out at you. And I've got my hand, at the end is my hand's fist, and as I come out and I go inside the eyeball itself, I go poof, I bloom on out, and now I'm covering the inside of the eyeball. And so that's what it's like. When your retina, when it's collapsed, is basically like the optic nerve, but now it's all together instead of being all fanned out for actual stimulation. So that's why we can liken it to an out pocket of the brain, because actually everything starts together and these little buds grow on out and floop, and open up and actually create the inner layer of our eyeballs. It's really fascinating to actually watch an embryological process. What we have closest to the pigmented layer and the choroid are our photoreceptors. We have rods and cones as our two photoreceptors, and there are 125 million per eye, so 250,000 total. The photoreceptors will then attach to bipolar cells, so bipolar neurons. So photoreceptors are at the back of our thin layer of our retina. And then the next layer is bipolar cells, and the very last layer, which is closest to the vitreous humor, are the ganglion cells. And so as we take a look at this histology drawing here, here are the nuclei of our photoreceptors. Here are the nuclei of our bipolars. And then here are the nuclei of our ganglion cells. So what's interesting about looking at a histology picture of a retina is it has very distinct layers, and the nuclei are what adds the really cool aspect of this distinct layering process. So they're all really close together and packed in there quite intensely. So photoreceptors very dense, bipolars a little less dense, and then ganglion cells even less dense as far as that goes. What's fascinating is take a look at this picture over here. It says the yellow light. Light is coming in to stimulate the photoreceptors and has to go through the ganglion cells and the bipolar cells in order to get to the photoreceptors. So it's important that you understand that that layer is, though you saw it as opaque, it is translucent. It is transparent, not translucent, it's transparent. You're able to actually transmit light through it. So it will stimulate the photoreceptors at its backside instead of its front side. And then the neural information is going to go in this opposite direction. So light's coming in one way, and the neural information going to your visual cortex in the occipital lobes of your brain is going in the opposite direction. A lot of things about the eye is kind of backwards from what you think. So always keep that in mind, that you're going to have to make yourself go backwards in what you originally want to first think, and that will actually save you quite a bit. The other thing too is axons are coming from our ganglion cells, and the axons are going to go to the optic disc, right? To the optic nerve and then into the optic tracts after the optic chiasma and stuff like that. Well, the axons, and I'm going to just act like the optic disc is this way, they're coming off and they're all going over here. So even the axons are kind of obstructing the path of light. Do you guys follow me? So a lot of this stuff that you actually saw, which made you think it's not so clear, it's fascinating to know the clarity of this layer is like the clarity of your cornea. It actually is very, very clear. And when people are looking inside the eyes, because it's so transparent, they're seeing other structures behind it, as well as vascular structures. So that's going over the surface. All right, optic disc, area where the optic nerve exits the eye. And with the optic disc, there are no photoreceptors here. And that's why we call that area the blind spot. That is the only attachment site of the retina, correct? At that optic disc. Now, the macula lutea and the fovea centralis belong to the retina. They do not belong to the choroid or anything else. They belong to the retina. Macula lutea, macula is Latin for spot, and lutea is yellow. And so as you take a look at a living eye, there's this yellow spot that you see, and the yellow spot is straight back. So the macula lutea is literally, as you take a look at somebody's eye, the macula lutea is straight back. And it is yellowish in its appearance, and in the very center of that is a spot called the fovea centralis. In the very center of the macula lutea is a spot called the fovea centralis. You cannot see it except with a microscope, but it's there. It's a very important spot because also in the fovea centralis there are no bipolars or ganglion cells located there. We'll go down here in the notes. Fovea centralis, pit in the center of the macula lutea, contains only cones, no bipolars.
Excuse me, I'm sorry.
That's all right. Or ganglion cells. So the ability of light to stimulate this area is even intensified. Can you guys follow me with that? We have nothing, even though it's transparent, we have nothing that's actually in the way of light to this spot. This is a cone-only spot. This is the area of absolute highest visual acuity. And it makes sense too, because it's literally straight back of straight back. It's like the bullseye on a target. That's our fovea centralis. The target itself is the macula lutea per se. Macula lutea are small oval spot, yellow, in a living subject. You cannot appreciate that in somebody that's passed away or preserved eye. And contains mostly cones. You notice that? Mostly cones. So is it all cones? No, it is mostly cones. So what else would be there besides cones? Rods. And so if you were to kind of draw a macula lutea, I'd say mostly cones. And then I'd have this little spot there called cones only. Now this is all part of the retina, so now I have more cones and some, I guess I should have done it a little differently. Here. Oh, still got my eraser. That really messed me up. More cones. How about some rods? And as we get further away from this, what do you think happens? We end up with more rods and less cones. So that's how the retina is constructed. Our notes also say that the macula lutea, and therefore the fovea centralis, is lateral to the blind spot. I want you guys to actually think about your sheep brain dissections and the optic nerve and the optic chiasm. So the optic chiasm is literally in the center of the brain, right, as far as the sagittal section goes, and then the optic nerves V out from that. Well, your eyeballs are not far from that. Okay, and so what happens is those optic nerves enter your eyeball nasally. You guys follow me with that? So the optic nerves are not very long, and remember we were just at a little X. They come out and they enter the eyeball nasally, and as you go lateral to the optic disc, you will find the macula lutea and fovea centralis. You guys follow that? Because if we were to look inside the eye to find the blind spot, we wouldn't look straight back, we would look nasally, because that's actually where it comes in. And so that's why in the notes it says lateral to the blind spot. Internal cavities and fluids, we've already discussed this, but let's go ahead and fill in the notes. Posterior cavity behind the lens filled with vitreous humor. Vitreous humor is also known as the vitreous body, a clear gel within that posterior cavity or segment, and that's why I wanted to introduce that word too. You might see it in some old exams or some notes. It supports the eye. Supports eye shape, plus keeps retina in place. Also supports posterior surface of our lens. So if you have a retinal detachment, that tells you right off the bat there's a bubble that occurred in the vitreous humor, correct? Because the vitreous humor, as long as it's nice and big and voluminous, is going to keep that retina where it belongs. But if there gets to be a little bubble in it, that retina can fill into that little defect. You guys follow me? And your vision is completely dependent upon the location of the retina. And when the retina moves, you no longer have that visual experience as the same visual experience. Also, that retina no longer gets support from the choroid. And part of its support is not just a little bit of the aqueous humor that seeps around the vitreous body. It needs its support from the choroid. So if you take the retina away from the choroid, you've taken it away from its major blood supply. And it too will also die. And so that's why they want to go in and they want to tack it back against the choroid and the sclera as soon as they can, and they'll do that, and hopefully everything will get reestablished and proper nutrient supply, and you won't lose your vision in that area where you had the detachment. Another thing I forgot to talk about was macular degeneration. You guys have heard about that, maybe? So macular degeneration is of the macula lutea. And does anybody know anybody with macular degeneration? Right.
Yeah, optometrist told my brother he had the markers for it, or he was predisposed to it. I don't know what that means though.
Yeah. Well, he saw some problems with health of the macula lutea. But if he wasn't experiencing any visual defects, then he's seeing some early signs of potential health-related issues. So the macula lutea literally is straight back, and if your macula lutea is sick and decaying and therefore dying, what vision do you lose? Everything's straight in front of you, right? So the macula lutea is straight back, and things that are straight in front of you are going to hit straight back. Because think of a dish, you know, a satellite dish. If something's off to the side here, where does it hit the dish? It can't go straight, right? If it's off to the side, it's going to hit it at these various angles. And indeed, that's what happens with our retina. When something's off to the side, that information comes and hits the retina off to the side, just like it would in any sort of a dish type of situation. So the macula lutea is straight back, and if people have macular degeneration, the things that they are looking at right in front of them, they cannot see. They can see things in the periphery or the perimeter, but they cannot see things straight in front of them. And so people who do have macular degeneration learn to compensate and never look at anything straight on. They have to look a little bit off in order to actually have other areas of their retina activated for the visual image. Anterior cavity divided into the two chambers, anterior chamber in front of the iris. In other words, the iris's dividing line, posterior chamber behind the iris, but in front of the lens. Aqueous humor is the watery fluid produced within that anterior cavity, and it is produced by capillaries of our ciliary processes 24/7. And therefore, if it's ciliary processes, it's in area of posterior chamber. Gets to the anterior chamber through the pupil and then is able to leave via the scleral venous sinus, the overflow aspect of things. So scleral venous sinus is located in the anterior chamber. And so this aqueous humor is going to supply nutrients as well as low level of oxygen to our lens and our cornea. And our lens will still get, because we have oxygen from the outside world able to get through, we'll still get oxygen from the outside world getting through our cornea into the area of our lens, and also providing nutrients for that. Again, here's a nice little picture of the scleral venous sinus. You'd actually have to cut through that area in order to actually identify that. But the scleral corneal junction is where you would actually go to find that.
What's cataract?
We're going to get into cataract too. So,
Okay.
Lens, and that's coming right on into it. Lens is a biconvex, transparent, flexible disc composed of lens fibers. So biconvex means in or out? Out. That's right, on both sides. That's what our lens is like. It's an outie on both sides. Not an innie, it's an outie. Protruding out on both sides. And it is also avascular. The bulk of the lens are lens fibers which are densely packed proteins called crystallins. So crystallins and lens fibers are the same thing. That's the name of the proteins. And they're layered in that lens like an onion. And so some of you actually did cut your lens in half and actually could appreciate some of that layering that was associated with the very firm ones. You could not do that with the frozen ones that were thawed out because they just started to splat in your fingers like a bug against a windshield. So the lens itself has a capsule, and when we look at various organs, like the liver, it has a capsule, the kidneys have capsules. Remember, the bone had periosteum, cartilage has perichondrium. So there's always this nice protective layer around very important structures and organs. So we have a capsule with the suspensory ligaments attached to the periphery, and this helps keep the lens in place. Tension on the ligaments will affect lens thickness. Okay, so the lens is a gelatinous structure. So let's go ahead and in our mind, think of it like a balloon filled up with Jell-O. Okay, and so we have these, and the balloon is the capsule and the Jell-O is the lens inside. And now we have suspensory ligaments attached to the perimeter of this lens capsule, and we pull on them. What will happen to the thickness of the lens? It's thinner. And then when we let go of the tension, it thickens. So that's what's so important. Our lens is pliable. And so we pull on the suspensory ligaments or ciliary zonules, and that will flatten the lens. And then we let go of the tension, and the lens will naturally want to go ahead, bloop, like a jelly belly kind of a thing, and just flop on out there and show itself off to the world. And this is important because we're coming now into the physiology of vision. And this is what helps us to see up close. Whether we can make our lens fat or thin. What do you suppose is better for seeing up close, a fat lens or a thin lens? Fat lens. Because when you want a magnifier to look at some detail of stuff, do you pick the one with a thinner lens or the thicker lens? Thicker. So it's the thickness that we can get to our lens, which allows us to see up close. One of the things that you have to realize, in order to survive in your world, it's not the things up close that are going to impact you unless you let them get up close. So your ability to see things in the distance is so important. So you can scan the world and you can make sure the world is safe for you. And so the stuff that we do, this reading and writing and stuff along that line, that's not a survival thing. You guys know that. It's not a survival thing. It's just helps you with certain daily tasks per se. But for survival, your lens is, unless you're trying to actually look at things up close, is always thin. So that tells you the tension on those suspensory ligaments or ciliary zonules is always tight. And then when you are looking at things up close, that tension must obviously relax so you can take a look at them up close. We're going to discuss how the ciliary body does that for you in just a moment. Before I do that, let me talk about the cataract question. Okay. When somebody has a cataract is an obstruction, causes an obstruction to your vision, correct? It is truly blocking the light rays from getting to your retina. What structures have we talked about so far can go from clear to opaque and therefore affect your vision? Cornea. As a matter of fact, you can have corneal opacities develop from like shingles and things like that. That's why shingles is bad for you, especially if you get ocular shingles or ocular herpes, because it can actually scar your cornea and then cause you to actually have difficulty seeing. So cornea can become clouded, which will affect your vision. Then what else can become clouded to affect your vision? Aqueous humor, but usually that's produced 24/7 and replaced, right? So the chance of that really becoming pathological and affecting your vision is pretty unusual. So then the next thing as we go on in is the lens, right? And that's what most people actually think about when they think of cataracts, is that the lens has now become opacified, is no longer transparent. And so something has caused it to become opacified. And what happens is as we age, and we're going to talk about this in a little bit, the lens becomes less flexible. Aren't we all less flexible as we age? The lens has that problem too. Even though it's got this lovely nutrient broth surrounding it, it with time, and light rays, photodegradation itself becomes less flexible. And because it becomes less flexible, it also will actually allow for less diffusion of nutrients into it. And when you get less diffusion of nutrients into it, you'll start to get opacities. You guys follow me with that? And the opacities, once it's like a pearl. Once you get started, it just seems to grow and grow and grow. And so the lens is a very common source of cataract formation. It's actually a more common source in our society. Another area that can actually become opacified that's supposed to be clear is the vitreous body or the vitreous humor. And so one of the things that's important about your vitreous body or your vitreous humor, you're born with it and you don't replace it. And so it too can actually become clouded. And some of you have animals that have developed clouded little looking pupils. They all look white. And I have a poodle and that passed away, and poodles very common will actually have genetic defects of their vitreous humor as time passes by. And so when you see those dogs that have like whited out pupils, more commonly it is actually some sort of genetic defect that actually caused their vitreous humor to become actually no longer clear but opacified. And you'll see that as reflected by looking at that. The important thing too about your retina, and this is why blindness is so bad early on, is that if your neurons are not stimulated, what do you need them for? If your neurons are not stimulated, they die. So that's an important phenomenon too. If somebody is born with a congenital cataract, they want to remove them as soon as possible, because if they don't remove that cataract as soon as possible, the retina is not stimulated, right? And the retina will die. Once the retina dies, it doesn't matter what you do to restore their vision after that, you will not get vision. So the retina, remember a lot of things, if you don't use it, you lose it. And same thing with our neurons of the eye. If they are not properly stimulated, they will actually start to die. And so this is another phenomenon to be aware of. The nice thing about the lens, because it's gelatinous and it's supported in a capsule and is suspended in its place, you can go in with cataract surgery, make a little slit in the cornea, and you can take your little suction and you can first off, you'll take your little knife and you'll make a slit in the capsule, and then you'll take your little suction device and you'll slurp that little lens out of its capsule. And then you have this bag left there, and then you take your implant and then you slip it on in that cut, and you slip it into the capsule, and then you stitch things up. You don't actually even do any stitching to the capsule because it will heal on its own, and then you actually have an intraocular implant. And so then they can see. But you've lost something when that implant is done, when that lens replacement is done, because it's solid. It is not pliable, it is not flexible, because they don't have the type of material that they need to actually create something as synonymous to our lens as we do. But when you can't see, taking whatever is out of your way, which is the lens in this case when a cataract forms, and putting in something that you can see through, that is the proper diameter or thickness to see your world, works. You guys follow me with that? And though you'll still need visual correction, and that visual correction will be for up close, because up close is something that we can easily make corrective lenses for, which is actually what I have these guys on for. So that capsule is actually a very important thing of allowing us to actually create this procedure and process. So lens is in place by the capsule and the suspensory ligaments, as well as our vitreous body. So when you get punched in the eye, you know, that lens gets a little roughened up, but that vitreous body's back there and it says, well, you're not going anyplace, so it keeps you right in place. It's your little airbag that's always been deployed. Take care of you. So now into the physiology of vision, refraction and focusing of light. Light rays are straight. They are wavelength, okay, but the waves follow a linear pattern. And they have to hit the retina. And if you want the best vision, you want them to hit the cones, because cones are for best vision. Rods are for night vision. Cones are for best vision, which is daylight vision. And in order to get in good light, in order to get something that we want to look at, to always hit that spot, if you're far away, your chances are pretty high you're going to hit that spot, correct? But the closer you get, if you're looking at something off and the area is a little off to the side, it's going to come in and it's going to go over here. Well, we want it over more in the center where our cones are in highest density. So we need things to bend that light ray to make it go in the area where we're going to get the best visual acuity. And that's what refraction is all about. Refraction is the bending of light. And we use lenses for that. If you do much photography, you have all kinds of lenses. You know, you have thick lenses for your camera, thinner lenses. You know, the thick ones are for macro and all that micro stuff. And you change lenses. But in we don't change lenses. We just change thickness of lenses so that we can actually see up close. So we can cause the light to bend by passing them through lenses which have curved surfaces, or we can pass it through clear media with differing densities. And so we take a look at this spoon over here in the glass cup that has water in it. When we take a look at this portion of the handle, we are only looking at the information coming to us from light passing through air, correct? When we take a look at things right here, we're taking a look at that information coming to us from air and glass, correct? And this glass is not causing much distortion in that vision. Maybe a little bit smaller, but not quite so much. And now here, we're looking at that spoon that's we're getting information still to us through air, because we're still looking at this picture here, or this object here. Air, glass, and water. And water has a different density, as well as glass does, from air, and we see this in a different size, correct? So this density of this media caused us to get a bigger image here. And that's what we do with some of our lenses. We make lenses out of differing materials or differing densities, which allow us to bend light more. And in addition to that, we can shape the edge of the lens to also help us bend light more. And we'll just go ahead and stop there and finish off this chapter next time, or this area next time, because vision is half of this chapter.